Namaste students, my name is JVS Dr. Neeraj Dhulia, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. So today we are going to discuss a topic that is molecular cloning, and this belongs to the subject microbial genomics. So in the molecular cloning, we clones the molecules. What are the molecules? Molecules are the genes or DNA fragments. So the population of exact copy of a or their production of a particular gene or DNA in the genetic engineering is known as the gene cloning. So the gene cloning, the recombinant DNA technology, the molecular cloning, they are more or less same thing. Okay, so when the DNA is extracted from an organism, all its genes are obtained, and in the gene or DNA cloning, a particular gene is copied from the clones. What are the clones? So clones are the uh, similar things, similar, or you can say the identical things. So cloning is one method that is used for isolation and amplification of gene of interest. So DNA cloning can be achieved by two methods. You can utilize the cell-based method of cloning. You can again you can uh, use the cell-free DNA cloning methods like the PCR. Okay, so here we will discuss the requirements of the gene cloning for cell-based methods. Okay, so in the cell-based method, you will need the DNA fragments that containing the desired genes to be cloned, then restriction enzymes and ligase enzymes. Then you will need the vectors to carry, maintain, and replicate the clone's genes in the host cell. Then you need the host cell in which the recombinant DNA can be replicated. So the fragment of DNA containing the gene to be cloned is inserted into the suitable vector first to produce a recombinant DNA molecule. The vector acts as a vehicle that transports the gene into a host cell, usually a bacterium, and although other types of living cells can be used. So within the host cells, the vector multiplies, producing the numerous identical copies not only of itself but also of the gene that it carries. So when the host cells divides, copies of the recombinant DNA molecules are passed to the progeny, and further vector replication takes place. After a large number of cell divisions, a colony or clone of identical host cells is produced, and each cell is a clone contains one or more copies of the recombinant DNA molecules. The gene carried by the recombinant molecule is now said to be clone. So there are some steps which is utilized, which is followed in the gene cloning. So first is the isolation of the gene or the DNA fragments of the clone. Then insertion of isolated DNA into a suitable vector to form the recombinant DNA. The introduction of the recombinant DNA into a suitable organism known as the host. Then selection of the transformed host cell and identification of the clone containing the gene of interest. Further, the multiplication, the expression of the introduced gene in the host cells, then isolation of multiple gene copies, protein expressed by the gene, and finally the purification of the isolated gene or copy. So, how to isolate the DNA fragment or gene? The target DNA or gene to be cloned must be first isolated. A gene of interest is a fragment of gene whose product insert interest us. For example, gene encoding for the hormone insulin. So insulin is needed for the diabetic patients. So the desired gene may be isolated by using restriction endonuclease RE enzymes, which cut the DNA at a specific recognition nucleotide sequence known as the restriction sites towards the inner region. So producing the blunt or sticky ends. Sometimes the reverse transcriptase enzymes may also be used, which synthesizes the complementary DNA strands of the desired gene using its mRNA. Then the second step is the selection of the suitable cloning vector. So the vector is carrier molecule, as we have discussed in the earlier lectures, which can carry the gene of interest into the host and replicate there along with the gene of interest, making its multiple copies. The cloning vectors are limited to the size of interest and that they can carry. Depending on the size and the application of the insert, the suitable vector is selected. The different types of vectors are available for cloning and plasmids, bacteriophages, bacterial artificial chromosome bags, yeast artificial chromosomes YAG, and mammalian artificial chromosomes MAX. However, the most commonly used cloning vectors include the plasmids and bacteriophages. Beside all the other available vectors are there. Then the essential characteristics of the cloning vector. So what qualities a cloning vector should have? So all the cloning vectors are carrier molecules, carrier DNA molecules. These carrier molecules should have some characteristics, like it must be self-replicating inside the host cell, because it is needed to be replicated with the host cell. So it must possesses a unique restriction site for the restriction endonuclease, so that it can be easily isolated, it can be easily ligated from one host to another. Then introduction of donor DNA fragment must not interfere with the replication property of the vector. So it should be separate from the host genome. It will not be interacting. Okay. It must possesses some marker gene, such as what the marker genes will do, it can be used for later identification of the recombinant cells. Okay, so it may be an antibiotic resistance gene or other genes. Okay, again the vector should be easily isolated from the host cells, means it should be easy enough, it should be handled easily. So the vector should have the mentioned qualities. Then the formation of a recombinant DNA. So the plasmid vector is cut upon by the same restriction enzyme used for the isolation of the donor DNA fragment. The mixture of the donor DNA fragment and the plasmid vectors are mixed together. In the presence of the DNA ligase, base pairing of donor DNA fragment and plasmid vectors occur. So the resulting DNA molecule is a hybrid of two DNA molecules, the gene of interest and the vector. So in the terminology of genetics, this intermixing of different DNA strands is known as the recombination. So the new hybrid molecule, DNA molecule, is also called a recombinant DNA molecule, and the technology is known as the recombinant DNA technology. Then transformation of the recombinant vector into the suitable host. So the recombinant vector is transformed from suitable host cell, mostly a bacterial cells, and this is done either for one or both of the following reasons. One is to replicate the recombinant DNA molecule in order to get the multiple copies of the gene of interest, and to allow the expression of the gene of interest that is produces its needed proteins product. Some bacteria are naturally transformable. They take up the recombinant vector automatically. For example, the Bacillus, Haemophilus, Helicobacter pylori, etc. Some other bacteria, on the other hand, require the incorporation by artificial methods such as the calcium ion treatment, the electroporation, etc. Next step is the isolation of the recombinant cells. The transformation processes generates a mixed population of transformed and non-transformed cells. So the selection process involves filtering the transformed host cells only. For isolation of the recombinant cell from non-recombinant cells, marker genes of the plasmid vector is employed. And for example, PBR322 plasmid vector contains different marker genes like the ampicillin resistant gene, tetracycline resistant gene. When PST1 restriction endonuclease is used, it knock out ampicillin resistant gene from the plasmid so that recombinant gene become sensitive to the ampicillin. The next one is the multiplication of the selected host cells. Once the transformed host cells are separated by the screening process, become necessary to provide them optimum parameters to grow and multiply. So in this step, the transformed host cells are introduced into a fresh culture media, and all these stages, the host cell divide and redivide along with the replication of the recombinant DNA carrier by them. So if the aim is to obtain the numerous copies of the gene of interest, then simply replication of the host cell is allowed, but for obtaining the product of interest, favorable conditions must be provided, such as the gene of interest in the vector expresses the product of the interest. Then next is the isolation and purification of the product. So in the next step, the isolation of the multiplied gene of interest attached with the vector or of the protein encoded by it. And so this is the followed by the purification and the isolation of the genome copy. So the applications of the gene cloning is a particular gene can be isolated and its nucleotide sequence may be determined. And control sequences of DNA can be identified and analyzed. The protein, enzyme, RNA function can be investigated using these. Mutations can be identified. For example, gene defects related to specific disease organism can be engineered for specific purposes. Again, example, insulin production and insect resistance may be produced using the molecular cloning. So these was all about the molecular cloning, and hope you are satisfied with this digital session. And if you have any query, you can write in the comment box. I will resolve that. And this session is powered by Digital Version 2, Jyoti Vidyapeeth Women's University, Jaipur. Thank you.